well, welcome to this afternoon's session. Hello, my name is Kirsten Matthews. I'm the Fellow in Science and Technology Policy at Rice University's Baker Institute for Public Policy. If you're not familiar with the Baker Institute, which I think most of you are, but we're a nonpartisan public policy think tank based at Rice University. We conduct policy research in several areas, including energy and environmental policy, international relations, taxes, which nobody likes, and health and medicine. My research specifically focuses on ethical and policy issues related to emerging technologies in biomedical research, as well as federal advice and funding for science, which is probably why I got to moderate today's session. It's really my pleasure to introduce our speakers for the health and medicine session. Over the past several decades, we've seen technology significantly change how science is actually being conducted in the lab. For example, back when the Human Genome Project was just in visions, researchers were actually using radiation to tag nucleotides, A, C, T, and G. They were following the sequences by hand. Now, not only is the technology allowing us to read longer strands of DNA from like a couple dozens to thousands, but we're doing it significantly faster and cheaper. The Human Genome Project took 13 years and cost estimated over $2 billion. Now it could be done for less than $1,000 a genome. And we're talking days or hours instead of years. This allows us to kind of collect thousands of genomes, to compare them, to identify differences between populations that might be associated with diseases. In addition to genomic data, we're also able to collect data on how healthcare and interventions are delivered, maybe link them to genetics or drug treatments, especially genetics and cancer, for instance. We're also collecting more and more data using wearable technologies that allow us to track biomedical data throughout the day, including your heart rate, maybe your sleeping patterns, if you're women, your menstrual cycle. Using data collected and technology collected and developed, we are able to begin understanding really more complex biological systems than we were before. How does regeneration work in the body? How does the early embryo development occur? How do neural networks form and create, these dynamic memories that we have? But what can we do with this data beyond just collecting it? How can we collect it and ensure patients' and individuals' privacies? How can we safely study groups of individuals to determine generalizable information if the data, for instance your DNA, is ultimately identifiable? How can we ensure that technology developed cannot be used for or manipulated to cause harm, such as intracranial devices used to record brain activities? How can we move from generalizable medical knowledge to help individuals with their specific needs? This idea of personalized medicine we've been promised since the human genome was completed in 2003. How can we ensure that research and ultimately new interventions are fairly and equitably accessible? Race and ethnicity are social constructs, but when research is conducted on a small number of genetic diverse individuals, how can that data really help others that are outside of that group? We already know that research using only male participants has limited our understanding of things like cardiovascular disease. It is very apparent that our data on other diseases and interventions might be limited to only a few race or genetic backgrounds. How can we ensure that this technology we're developing is accessible financially to everyone? The United States already has a high cost of health insurance, and it's growing. Private and public medical insurance don't cover all people, especially in Texas, that has the highest uninsured rate in the United States at 18%, which is double the US average. This afternoon's session will focus on several of these aspects of how medical research and health policy is changing as a result of increased use of data. We have three fantastic speakers, each discussing their area of expertise in ethical, legal, and social questions related to the research, either that creates or addresses. They will each focus on a distinct area of their research concerns. We're going to start with our engineer, Dr. Behnaam Aazhang. He is the G.S. Abercrombie Professor of Electrical and Computer Engineering and Director of Rice University's Neuroengineering Initiative. Dr. Aazhang will review his research on neurological disorders, especially epilepsy, a set of neurological disorders that cause brain seizures. His work focuses on understanding, detecting, and predicting seizures. This work requires understanding of how neurons connect with each other, using and developing technology to monitor how they communicate with each other, identifying signs that something abnormal has occurred or will occur, and finding ways to treat abnormalities in the least invasive way possible. His group is working on developing devices that could predict seizures to allow patients to stop risky behavior, such as driving. You don't want somebody to be driving and having an episode, and seek medical help, as well as creating implantable devices that could perhaps even preemptively stimulate the brain to prevent seizures from occurring. Today, he will review a lot of his work in neural engineering, the challenges researchers face, and some recent work his group has done to address them. Our second speaker will be Vivian Ho from she's the James A. Baker III Institute Chair in Health Economics at the Baker Institute for Public Policy and Professor of Economics at Rice University. Dr. Ho is a renowned expert in health economics and U.S. healthcare policy. She has a long history of studying the cost of medical interventions, insurance, emergency care clinics, and hospital costs. Dr. Ho's work also examines the effects of economic interventions and regulations on the quality of and the cost of healthcare, including Affordable Care Act. For her presentation, she will review the wealth of data accessible related to the cost of healthcare, focusing on the cost of emerging and novel healthcare technologies, how they impact overall healthcare costs, and how regulations can be used to control those costs while still encouraging innovation. And finally, our third speaker will be Dr. Amy McGuire, the Leon Jaworski Professor of Biomedical Ethics and Director of the Center for Medical Ethics and Health Policy at Baylor College of Medicine, which is in the Texas Medical Center across the street. Dr. McGuire's research focuses predominantly on ethical, legal, and social implications of genomic and genetic research and data. With the sequencing of the human genome in 2003, we have seen an explosion of research sequencing genomes, analyzing the data, and storing them. We have also seen emergence of new companies offering sequencing technology to anyone marketed to help identify maybe your heritage, disease risks, or even sometimes taste preferences. Dr. McGuire's work dives into questions that emerge with this large collection of data, including improving informed consent by ensuring patients or clients understand how their genetic information is being used, determining when and if patients should be informed of genetic risks found during incidental research findings, identifying policy and regulatory gaps, especially in direct-to-consumer genetic testing market, and ascertaining public ethical concerns as well as policy challenge with the research in general. This afternoon, Dr. McGuire will explore how direct-to-consumer genetic testing is being used, ethical concerns that emerge from it, policy implications, and common misconceptions, all based on the work she's been doing for over a decade. From these three presentations, we hope to share with you some of the challenges facing use of data in health and medicine. We also want to highlight the opportunities to understand health and to develop novel technologies that this data and technology allows for. So I think the next step is to get it started with our first speaker. I want to welcome him to the stage, Dr. Aazhang, to give his talk entitled Engineering in Medicine, Learning, Inference, and Control.
She summarized so well that I don't know what to say to add to what she said.
You say, as she said.
As she said. Is this working? Can you hear me? Yeah. Perfect. You are? All right. The title is appropriate. I'm going to talk about learning from data, as she said, and I'm going to talk about inference, what can we learn from the data, and control. How can we take an organ like the brain and move it from an unhealthy state to a healthy state. That's the agenda for today. Before that, I want to brag about my colleagues, our team, and these are the team members, and we are part of what we call Neuroengineering Initiative. It is a little different from neuroscience. We're a bunch of engineers. And as you know, engineers think they can fix anything. But don't tell my wife that. We like to fix things. And one of the things that we like to fix in this context is the functionality of our brain. All right? And as you could imagine, it's not an easy problem. And if you work by yourself as a bunch of engineers, you're wasting your time. You got to work with clinicians. And this is the sort of list of clinicians across the street, literally, that we work with. These are my colleagues from Baylor College of Medicine. And I'm going to talk about some of our work with these three individuals. And it's going to be fun. And I also work with folks at Houston Methodist. Houston Methodist is an incredible hospital, and it has a research institute. And I am going to be talking about our work with both research institute and clinicians. And lastly, I want to talk about our work with UT Health. UT Health is also literally across the street. And in that, I work with Nitin Tandon, who is a neurosurgeon. Surprisingly, I don't work very much with neuroscientists across the street. I work with neurosurgeons. Don't know why, but somehow it worked out. All right? And we're going to talk about this. This is supposedly our brain, is a depiction of our brain. And as you can see, there are a lot of neurons, hundreds of billions of neurons, and they're all connected to each other. All right? And you can look at this thing and say, okay, it's very complicated. It looks like internet, but is our brain. And it does a lot of different things. As you can imagine, if I try to remember Tony's name, a lot of things happen here, and then I mention his name, right? And if I move my arm, a lot of activities happened in my brain that allowed me to move my hand. It's very complex, and you can look at it at multiple levels. You can look at it at the neuronal level, at the cell level, and this neuron is going to send a pulse to this other neuron over there, and that it would be the start of their interaction, and then their interaction goes down to peripheral system and makes my muscle move. All right? This is basically what's going on. And you can look and study our brain for years and years and years at the cell level. All right? That's not what I do. I have no idea what it is and how it works, but that would be a neurobiological aspect of this work at a very low level. What I do, because I'm an engineer and I know very little thing about neurobiology, I work at this level. I work at the system level. Basically, what is going on here is that we start out by recording. We record from the brain, and a whole bunch of different recording channels over a long period of time. And then we take those recordings and we learn from them, both about the circuit underlying that recording, and also try to learn what is it trying to do, all right? And then turn that learning and turn that inference that we make from that into control. Basically, you could imagine that control would be giving a signal to the brain, telling it not to do this, do that instead, all right? And that control, I'm going to talk about that today, and then that control, then we can watch what happened. We can, again, look at the action to see whether this movement of my arm happened or not, and then record again, and then close the loop. All right? This is basically what's going on. And these are some typical recordings that we work with. In this case, I am depicting here 30 channels, meaning that 30 different recording spots, and then across 9 minutes, let's say. Right? And these are the kind of signals that we work with, and these are the kind of signals that we're trying to learn from. Right? And that's the genesis of our work, at least my work. My colleagues do very different things, but this is the kind of things that I do. All right? Now, why are we doing this thing? Because there are a number of disorders that exist, and we're trying to learn about them. All right? I can list hundreds of disorders, brain disorders, and I just mentioned 3 of them here, which are very, very prevalent, and especially at older age, like my age, and epilepsy, Parkinson's, Alzheimer's, and all that. The list keeps going on. And I looked it up on Wikipedia, the source of all information, and the top 5 neurological disorders, this is 3 of the 5. All right? There are quite a few of them anyway. Now, today I'm going to talk about epilepsy. All right? epilepsy is an interesting phenomenon. I am fascinated with epilepsy, and I've been working in this area for 10 years, over 10 years, all right? And we don't know much, all right? What's going on is that 50 years ago, we didn't know what we don't know. We thought we knew about the brain, but we had no idea. Nowadays, we know what we don't know, which is kind of an interesting term, right? We know what we don't know, which is a step forward from not knowing what we don't know. And this is where we are. We really honestly don't know what we don't know. And what is epilepsy? Epilepsy is defined in Wikipedia as unprovoked and recurring seizures. Make sense? Basically, there are people who have 15 seizures a day. And these seizures are quite aggressive, and they cannot do much because they're expecting to have 15 seizures a day, roughly. And it turns out that epilepsy is defined this way, but the question is, what's the definition of seizure? All right? Again, that's not a standard. Even Wikipedia admits that the definition of seizure is not a standard. Basically, seizure could be defined as abnormally hyper-excited state. Basically, your neurons in a particular region get hyper-excited, all right? If it happens in motor cortex, you may shake, and you may fall down, and then it may happen in your language region. You may not be able to talk for a few seconds. Therefore, there are a variety of different seizures out there, all right? I was very surprised when I learned that 1% of world population suffer from epilepsy. I don't know whether you knew that or not, but it's a surprise. It's a large number. It's a large number. And it turns out that 1/3 of the patients with epilepsy are not responding to medication, all right? And that is termed as a refractory seizure, refractory epilepsy, all right? 1/3, that's again a large number. That means that they have seizures a lot, and there's not much doctors can do for them from a pharmaceutical perspective, all right? And the question is that what else can we do for those patients, all right? One idea is resection. As you know, surgeons love to do surgery, and what they suggest is that if the epilepsy is very local, they go in the brain and then they resect that region of the brain. And as you all know, a region of the brain doesn't do just one thing, is involved in maybe 10 different functionalities. Therefore, if they resect that region, you may lose 10 different functionalities, which is not a good thing to do, and we try to avoid that, all right? Another option is deep brain stimulation, that they put electrodes in the brain, insert the electrode down to the region that is supposedly epileptic or has frequent seizures, and then they stimulate that area 24/7, and there are a few products out there, and hope for the best. They really don't know what they're doing, but they do it, and they hope for the best, all right? Now, the question is, what else can we do? You know, and that is where we come in. As engineers, we come in, I don't want to say that, but we come in to save the day, basically. I'm just kidding. Basically, this is the story I'm going to tell about what can we do about seizures, how can we learn about it, and what can we do to help, all right? Now, what I'm showing here is 30 channels, basically 30 locations on the brain. I don't know whether you can see, yeah, you can see that. These are the electrodes that have been placed underneath the skull, on top of the brain, on top of dura, and there are some 200 electrodes that record from 200 different places in the brain, and the recordings could happen for days and days and days. And the question is, what can we learn from those recordings, all right? And the answer is, this is 30 channels of that 200 channels that we are going to focus on for now, all right? Again, if I tell you that this area from 0 to 4.5 or so seems to be normal, right? Seems to be normal activity of the brain that a lot of neurosurgeons, neurologists see it all the time, right? If I tell you that the patient went through seizure between 4.5 and 6 minutes, I have a feeling you believe me, all right? That there was a seizure that happened between these two black lines, all right? Again, this is something that neurosurgeons and neurologists tell me that this is a seizure area, all right? And one question would be, can we detect seizure with this technology, with these electrodes and all that? And the answer is probably yes. It doesn't sound too hard to be able to classify and detect this region from that region, to say that now you're having a seizure. I bet you the patient can tell you, yes, I'm having a seizure, but we can also tell the patient you're having a seizure, right? The question that is a million-dollar question is that, can we predict the onset of seizure? That would be a good thing. What does that mean? That means that the patient could brace himself from falling down. The patient could expect the seizure, maybe stop driving, pull to the side of the road, and many, many other good things that Kirsten mentioned, all right? And the question is that from these 30 signals, 30 channels, can we predict that there's going to be a seizure happening in a few minutes? So I think it's a good question. It's a good engineering problem to have, all right? And the second thing to say is, or the third thing to say, is that if you know the seizure is happening in a minute, can we do something about it? Can we sort of impose some signal, modulate or stimulate to prevent the onset of seizure? That would be another very good thing that we could do, all right? Again, these are the challenges we are facing, and I'm sharing those challenges with you, all right? Therefore, our approach is that we want our approach to be patient-specific. Therefore, a given patient, we don't want to just give one drug to everybody. We want the solution to be patient-specific. That means that we want to put electrodes and then learn about the seizure that that patient has, and then learn from that, and then try to predict the next seizure, right? Now, if the seizure is global, meaning that the whole brain goes through seizure, we are out of luck. And if the seizure is focal for that patient, that means that a small region goes through epilepsy seizure, and if the patient has the seizure in the same spot every time, then we are in business. But then if it changes, one day he has a seizure in this spot, next day he has a seizure in the other spot, again, we are out of luck, because there's not much we can do for that particular patient. It turns out that many, many patients have seizure in the same spot, all right? And also, the number of patients with global seizure is very limited, but the number of patients with focal seizure is very large. Therefore, there is hope for us. All right? And as I said before, we want to identify the seizure zone, but maybe more importantly, we want to identify seizure onset zone. We want to learn that if this electrode and the population of neurons underneath that electrode subscribe other neurons to get into the seizure mode. It would be nice to stop that, basically. That's the idea. All right? And then lastly, we want to modulate the brain to prevent the onset of seizure. All right? Now, this kind of work that I'm talking about now has been already published. If you're interested in more detail, you can look my name up in all these online data sets, and then look me up, and then seizure prediction and all that. You can find the article that we wrote about these kind of things. All right? Now, again, I'm showing you 3 channels here, the green, the red, and the blue. Again, if I tell you that the green channel is not going through seizure, you probably believe me. If I tell you that the red and blue channels are going through a seizure in a minute or two, again, it's likely that you're going to believe me, because it looks like they're going through a hyper-excited state. Right? Therefore, you can imagine that from the 250 channels that we're getting information from, it sounds like a few of them are going to be in seizure zone. All right? And the technology that we used for that to detect what channels are going to go through seizure is basically called graphical signal processing. It is signal processing. It's graphical because we have multiple channels, therefore multiple locations. And we developed this thing a while ago, and basically you're able to identify 10 to 15 channels out of 250 that are going through seizure. And the next question that I was going to talk to you about was identifying what we call seizure onset zone. That means that the channel or the region that starts the seizure and spreads to other channels. All right? And you can imagine that if I tell you that the red is the seizure onset zone, and it subscribes the blue to go into seizure, and then it kind of spreads. All right? Therefore, the red is what we call seizure onset zone. And the point is that if we can identify seizure onset zone, maybe we can go down from 250 electrodes or 250 channels to 3 to 5 channels that start the seizure. Therefore, we can focus our activities and our analysis on those smaller number of channels. Right? And the technology that we use for that is called the information theoretic method to identify causality. All right? What is causality? Causality is basically one event causing another. Let's say, for instance, if stock market goes down or crashes in China, we can imagine that it's going to crash in the US the next day. That's called causality, that the failure or the stock market failing is going to cause stock market failing in the US a day later. That's called causality. Granger got a Nobel Prize on that many years ago, developing the theory of causality, and it's called Granger causality. And what we have done is that we have looked at it from an information theoretic point of view, and we developed a causality measure to find out that the red actually does cause the blue to go into seizure. All right? Now, basically, we are down to 3 to 5, 3 to 6 or so electrodes that cause seizure. Now, the question then is, now with all of these things, we can focus on a smaller region and see if we can predict the onset of seizure. All right? That's where we are. And it turns out that this is another depiction of the same problem, that you could imagine that these patients went through a seizure starting at this point, let's say, and then we have time recordings of that patient here and there and all that. And the question is that, can you look at the dynamics of these recordings to predict the onset of seizure? And that is all about the dynamics of the signals across those channels. And there are just a few channels, if you remember. We went down from 250 to 3 to 5. All right? It turns out that we can. Again, we published all these things earlier, and we can predict with 95% accuracy the onset of seizure. 1 minute, 3 minutes, 5 minutes. You can imagine that the 1 minute is better than 5 minutes, better than 10 minutes, and all that, right? Now, the theory that we use in that is called embedded dynamic mode decomposition. Basically, what it does is that because of the fact that we're down to a few channels, it embeds those channels to get much larger matrix, and then uses the dynamics of that to predict the seizure. Right? And as I said, we have done it very, very successfully. The question is that what can we do next? Now that we know that we can predict the onset of seizure, and we have electrodes placed underneath the skull, can we modulate? Can we modulate and prevent the onset of seizure? And the answer is we don't know. All right? That was a good answer. And the idea is there, though. The idea is there that we wanted to be able to modulate the circuit to prevent the onset of seizure. All right? Let me back up and talk about electromagnetic waves. All right? I know that this is a very broad audience with a broad background. This is electromagnetic wave at gigahertz scale. That means that the frequency of it is gigahertz. All right? Now, our cell phone generates this kind of signals at 1.8, 1.9 gigahertz. And this is 2 point, my student draw this thing is like 4 gigahertz, let's say, for now. All right? The time scale you see is very narrow, therefore these are very high frequencies. All right? Now the question is that if our brain tissues are subjected to this kind of signals, is that going to impact or is that going to change the behavior of neurons? The answer is no. The reason is that these frequencies are too high and the ions are not going to move as fast as this, therefore they're just going to ignore the signal. That's why we are allowed to carry cell phones, we are allowed to have cell phone antennas generating signals at this frequency, because this frequency does not disturb our neurons. All right? Which is good news. We can use cell phones safely. The question is that how about if we really decrease the frequency to subhertz? The other one was gigahertz. This is subhertz. The question is that does this impact our neurons? The answer is yes, it does. What it does, most likely, is that it's going to inhibit our neurons to spike. Inhibit. Hang on to that for now. It's going to inhibit our neurons from firing. All right? Is it the good news? Maybe. We'll see. How about 120 hertz? Something in between subhertz and gigahertz. It looks something like this. It turns out that this has been proven that this kind of a frequency, signal at that kind of frequency, it actually excites the neurons. All right? Therefore, this kind of frequency at that range excites neurons. It allows the ions to work according to this frequency and vibrate and generate a spike and all of that. All right? Therefore, this electromagnetic wave at this frequency could excite the neuron. Hang on to that as well. The question is that how can we generate, let's say if we want to inhibit neurons from firing, inhibit them from firing, meaning that we don't want our brain to go through hyperexcited state, can we do that? Can we generate a subhertz frequency without disturbing the other part of the brain? All right? And can we do that? All right? These are my colleagues, and I work very closely with them in this particular technology. I just want to acknowledge them. All right? The last two are our students, and the first three are my colleagues that you saw their pictures earlier. I hope you remember them. All right? And these are the disorders that we could trigger, we could work on because of the fact that we have technology to inhibit and technology to excite neurons. All right? This is the idea. All right? Imagine that you have 4 or 5 or 6 electrodes that generate electromagnetic signals, and they create a beam because there are multiple of them, and just like a cell phone, they do not spread their signals. They focus a beam and create a beam that could penetrate through the brain. And because of the fact that this electromagnetic wave at that particular frequency, if that particular frequency is 4 gigahertz, is not going to disturb the neurons along the way. That's what we agreed on, right? It's not going to disturb the neuron along the way. Therefore, these 4 electrodes are going to generate signals, create a beam, and the beam is going to penetrate into the brain, but it's not going to disturb all the neurons along the way. All right? Imagine we put another set of electrodes, the blue ones, that are slightly offset in terms of frequency with the one in green. All right? And if this one is 4 gigahertz, the blue one is 4 gigahertz plus, let's say, what did I say here? Plus 100 hertz or plus 1 hertz. All right? Now, this is where an equation comes in, that these two beams, when they overlap, the signal is going to look like an envelope. And it's going to be just like that, the red one. because of the fact that these two signals are slightly different from each other in terms of frequency, the equations are going to dictate, the algebra is going to dictate that in this red region, when these two beams interact, you're going to have an envelope in red here, which has a much smaller frequency, meaning just about 100 hertz, let's say. All right? Now, here's the idea. Beam that penetrates does not affect the neurons. Beam that penetrates, the blue one, that does not affect the neurons, but at their intersection, which is a very focal area, is going to generate this envelope that could affect neurons. That's the magic, basically. Am I okay with time? All right. And because of the fact that these are beams, we have a lot of degrees of freedom. We can make the focal point here. We can make focal point here, focal point there. And depending on where the seizure is happening, or that particular patient has that kind of seizure, we could change and make the focal point at various different locations. All right? And the point is that this red stars are going to be the places that the frequency is going to be at the range that is going to impact the neurons. All right? Now the question is that going to work? The short answer is we don't know. And the long answer you're going to hear, the long answer is that we are optimists. We are researchers, we are engineers. We can fix anything, right? We have done a lot of simulations and identified all the different locations that we could modulate. We have done electromagnetic wave type analysis and then look at the heat, see how much heat does this generate, right? It turns out that if you want to get FDA approval, eventually, not now, eventually you cannot heat up the neurons more than 1 centigrade. 1 centigrade is the limit. If you can develop a technology that increases the temperature less than 1 centigrade, you're okay. But above that, you're not going to get FDA approval. And we've done all the analysis, and it is going to heat up less than 1 centigrade, right? Which is good. And then we have done phantom experiment. That means that we put electrodes, and then we had a petri dish with water and salt and all that, and then we checked to see whether we do get waves and all that. We got all of that done. And then there is another set of tools called biophysical tools, theoretical biophysics, basically, that looks and analyzes how neurons behave at the neural level. And we are working on that now to see how these waves impact neurons at the neuronal level. We don't have answers yet, but we're working on that. And the last one would be to do animal models, meaning that hydra, rodents, pigs, sheep, and all of that. If we can investigate this technology with those animals that are epileptic, that you can make them epileptic and then try to prevent the seizure, that would be fantastic, right? We are not there yet. We are working on that. And we are working on biophysical model. I didn't want to tell a story that has been done 2 years ago. I want to also tell you something that we are doing right now so that I can share my excitement with you, all right? And I hope I've done that. And the question then is, if we have the technology, what are what are our goals? What are some things that we could help? All right. We talked about epilepsy already, maybe too much. Can we help with depression or OCD? You could imagine that if you can excite a population of neurons, maybe the patient would come out of depression, maybe. And then we know, in principle, how to excite a neuron. We are not there yet, but in principle, we have an idea. All right? And we're working on that, and we're working on that, and we're also working on stroke. You can imagine that acute stroke, you may be able to excite the neurons to come out of stroke and start having some activities. Therefore, if we can simulate at 120 hertz, we may be able to bring those population of neurons out of their depressed state or out of their stroke, acute stroke, and then let them fire up and get excited. Same thing with glymphatic flow. Therefore, if there is a patient that has low glymphatic flow, that means that the patient had a traumatic injury or some other situation. Maybe we could help that patient by doing modulation and enhance their glymphatic flow. We don't know. These are the kind of projects we're working on right now, and that's it. That's all I got. I guess I can take questions or not.
So epilepsy seems like electrical problem.
Yes.
It makes sense to have an electrician or electrical engineer deal with it. But you're talking about like when you have a stroke, it starts with usually with a blood flow problem, right? Something
Yes.
has a blockage.
Yes, that would be one reason.
And my understanding is the neuron dies for lack of blood. And so I don't see why electricity would be a way to address it. Same thing we're talking about Alzheimer. It's electrical problems. Why would I have electrician? If I have a plumbing problem, I get a plumber, not an electrician.
Well, the plumbers cannot help with any of these things, right? But the idea is that these kind of stimulation may excite the neurons to help themselves. That's all the idea that we have. That the neurons, as I said carefully, is not stroke, is an acute stroke. Basically, a stroke that just happened. So that you could, before the neurons die, because of lack of blood, as they are disturbed, but they're not dead yet, we may be able to revive them, because as you know, our brain is dynamic. It changes all the time. We're going to help the brains to change, help the neurons to change, to improve. Did I answer your question? Again, as I said, probably 5 times already, we don't know, right? we are trying. And I appreciate your question about plumbing versus engineers. We don't know anything about plumbing. All we know is electricity. And we're thinking that if we can excite, that may help.
Very exciting.
Yes,
Hi.
sir.
Thank you so much. I think this is really cool work with regards to like reading and writing neuroactivity in the context of like therapeutics. I'm curious because in the future we might see applications of these tools in a non-therapeutic context, for example, like reading or writing to memory or thoughts. So I'm curious what your thoughts are on the implications of privacy and maybe autonomy with regards to the use of these tools in those types of contexts that might happen in the future.
That's a tough question. I don't know anything about ethics. I don't have it myself. But the ethical questions of anything that people do these days is an important question that I don't know and I don't have an answer for that, but it's important. Like how could you, basically help your brain learn languages better? That's basically disturbing a function that is supposed to be all natural, right? I don't have an answer to that. All we are focused on is to try to see if we can develop technologies to help people that have issues, not just to make them smarter. That is way outside my realm of interest or capability. But I appreciate the question.
Thank you.
Mm-hmm. Rick?
I want to get a little bit into the weeds because I don't quite understand the technique that you're using. So I assume it's like an EEG that you're using, which may go to a centimeter deep into the skull in terms of readings, or are you using something else?
It is what we call electromagnetic wave.
Ah, okay.
Electromagnetic wave, very much like what our Wi-Fi system uses or our cell phones uses and all that. It's just that because of the fact that it is going to be narrow beam and it's going to be underneath the skull or maybe on top of the skull, it's going to have enough energy to inhibit or excite neurons. Right now, when you carry your cell phone or you get signals from the same kind of signals from tower, cell tower, by the time it gets to your brain or inside the skull, it's such a low energy that doesn't do anything. What we're thinking is, what if we do that with high energy so that we can make a change?
So I think I phrased the question wrong. I was wondering on the measurement that you had of the 200 plus channels. Is this EEG or?
No, that's called ECOG.
Okay.
EEG would be another alternative, which is often or always outside the skull. ECOG, which I showed in my slides, are underneath the skull, very much like EEG, but they're underneath the skull, and they're similar kind of electrodes. They basically record
how far
signals.
down does it go?
Say again?
How far into the brain does ECOG go?
ECOG, in terms of the placement, is on top of the brain tissue, is not inside. And in terms of how deep can it measure from deep inside the brain, it all depends on signal-to-noise ratio. The deeper, the larger the noise and all that. And we have worked with signals that were epileptic, were a result of seizures all the way down to hippocampus, which is quite down.
Okay. So we can talk later.
Absolutely.
I was really interested in that part of the measurement. So
absolutely, absolutely. And ECOG is quite common, but as you can imagine, you have to be have serious condition to allow the surgeon to put electrodes underneath your skull. Therefore, that has to be an epileptic patient that is really suffering. Yes?
Kind of along that topic, you mentioned current existing technologies. One of them is, like, for example, DBS, deep brain stimulation, which is invasive, which can be risky with regards to surgical complications and perhaps like tissue damage, right? Which we don't want in the brain, which cannot easily regenerate. And so I was curious if there is potential for this method to be non-invasive because we're using like waves, right? Maybe in the future, perhaps going like through the skull and somehow still having it work in terms of reading and or writing.
Absolutely. I don't know why you're obsessed with reading and writing, but that's fine. In the ideal world, you would like to put electrodes on the skull, like EEG that he mentioned, and generate enough signals that would penetrate the skull, which is often quite dispersive, and and then create a beam that is narrow, so that you could focus on one area. All we have done so far is that we assume that the electrodes are on top of the brain, but underneath the skull, so that the problem would be simpler. But as you could imagine, and you said it yourself, that would be much more popular if it was non-invasive, meaning outside the brain. But it's a much harder problem. The question is that can we generate enough signals outside the skull so that it would penetrate and not burn your skin? We don't know. We'll find out. Yeah. Actually, that is something that we are interested in it ourselves, and is a next step. As I said several times already, this is very recent work. We have so many problems to tackle, and there are so many diseases that we would like to tackle. Read and write is not one of them, but it is the very, very next step.
Thank you.
Behnaam, thank you for a terrific talk. I have a three-part question. One, how much advance warning is there for the epileptic seizure? Like, what's the time scale of that? Two,
can I answer the first one?
Yeah, of course.
We have results that says we can do it in 5 minutes with let's say 80% accuracy. I'm making up numbers here. And then we can do it, let's say 3 minutes with 85%, and we can do it 1 minute ahead of time with 96%. And again, I'm making these numbers up because I don't remember, but not because of Alzheimer's, but yes, it all depends, right? It all depends. But we have done some analysis that showed that 5 minutes before the seizure was a pretty decent number.
How do you focus the beam or aim the beam accurately enough so you're not accidentally
Yes.
damping something that you wouldn't want to?
A very, very good question. It is a challenge between focality and intensity. This is an age-old question, right? How focal you want to be and how much you want to provide intensity to be able to do something, right? This is an optimization problem. And we have simulated it all day and all that. And we have some numbers. We have some area numbers as to focality areas. We have intensity numbers. And all of these have been published.
And then final question, how does your lab do the research into this? I mean, is it with human subjects? Is it computer modeling? How as you're trying to figure this out, do you work?
We have a lot of data thanks to our collaborators. I don't have any data ourselves. We don't have any data ourselves. Our collaborator has tons of data, and he is at UT Health, and he is very much an expert in epilepsy, and we are at the mercy of his data. And as you could imagine, there are a lot of data sets out there that we've worked on as well. It's just that your own data set gives you an edge that we can control. We know what we're doing. We know what the data is like. Therefore, we have a lot of data that shows the recordings of 24 hours or longer, and then the seizure has been identified by neurosurgeon. Now, this is a seizure, and then my students analyze the data, they identify the seizure, as the doctor said, and then try to predict the next one. Again, the doctor said there's a seizure here, therefore they see if they can predict it. Yeah.
And when you're coming to actually testing the cure, let's say, how will you do that?
Animal models. Animal models. As I said, mice, sheep, yeah. As you can imagine, animal models are very expensive. And especially larger animals are very, very expensive. But you got to do. Yeah.
I have a question about service animals. That I saw a video last year of a dog that this lady had who accurately predicted a seizure that she was about to have, and he ran up to her. She got off the sofa, and he ran up to her and pushed her back on the sofa, and she did indeed have an epileptic seizure within minutes of him encountering her. Can you tell me more about that type of...
I have seen the similar kind of videos. I am amazed, but at the same time, we have like 5 dogs. I can imagine that some dogs could be trained to do this thing, and their intuition, their sense of smell, is incredible. And other than that, I have no answer. I don't know if anybody does. I don't, for sure. But I am as fascinated as you are, and I've seen the same kind of videos and all that. Yeah. Very good question. I wish I knew that much better, but I don't.
Okay, thank you.
I just have 2 questions. So just to understand this better, I'm assuming that you do the electrodes and pinpointing the area so that this is a more permanent kind of solution, but how frequently do you expect to have the patient go under this procedure for it to be effective?
You mean the procedure, you mean placing the electrodes or activating the modulation?
Activating the modulation.
Oh, what we have in mind is that the electrodes would record continuously, and then our algorithms, which is probably sit somewhere here in some box here, would look through the data, just like the dog that she mentioned, would predict the onset of seizure, and then we would start modulation then. Yeah.
Okay.
And as I said earlier, that would only work if the seizure was focal and the seizure of that particular patient was always at the same place. so that we could use our experience from previous seizures to create beams and focally modulate. As I said before, if the seizure changes, we're out of luck.
Yeah. So for based off of that, like how many times does the seizure have to happen for you to measure and accurately predict that it is a focal and it's the same place? Because like how many times would you say would be able for you to say that, oh, it is focal, it is the same spot, so that there's not like unnecessary damage that occurs?
No,
if
no,
it isn't?
I understand. Very good question. A lot of times we rely on neurosurgeons. A lot of times. Because the electrodes that I showed you, they're placed on the brain. They could be placed here, they could be placed there, and all that. The neurosurgeons would tell us this particular patient, because of the fact that their seizure was creating a lot of movements for them, therefore this must be in the sort of motor cortex area. Therefore, they tell us where to place the electrodes or where to focus. And we live that. And other than that, the brain is, for human brain, is just too big for us to know and place electrodes everywhere. That's just too much work. Therefore, we rely heavily on our clinical colleagues.
Thank you.
Sure. That was your second question? Yeah. All right. Okay. All right, thank you.